Michael Tene, second-year graduate student at Lehigh University, and I'm excited to speak to you all today. What I'm interested in is connecting genes to phenotypes. While we understand the cellular function of many genes, how those genes contribute to overall organismal-level phenotypes is not as well understood. However, genes can be connected with phenotypes through their shared evolution. Obviously, trait and phenotype changes are caused by underlying genetic changes, and so by seeing which genes evolve alongside those phenotypic changes, we can connect genes and their genetic changes to phenotypes. This is difficult to do in individual species because you can't tell which genes are related to which traits the species have. This is where convergent evolution is incredibly useful. By examining the breadth of an entire phenotype, you can connect genes with the phenotype as a whole, as opposed to being in individuals. This is because convergent evolution provides natural replicates, where we see the same genes evolving separate times in different species. So, for instance, if we see something evolving in fishing bats, but not fruit bats, orcas, but not hippos, and polar bears, but not panda bears, we can be fairly confident that that signal is due to being a carnivore, as opposed to being, say, an orca. With alignments and a phenotype definition across a phylogenetic tree, this principle can be applied to any phenotype, and that wide applicability will come into play as a big part of my work. The phenotype which we have chosen to examine is carnivory, as opposed to herbivory. We've done this because diet is a convergent and complex trait with a wide variety of selective pressures that it imposes upon individuals. Some are pretty obvious, broad changes that might jump to mind with different ones depending on your specialization. Morphological changes such as tooth shape, behavioral changes such as food acquisition strategy, and general life history changes. The most direct change is a difference in food composition, with specifics, for example, being a percentage of peptides in the diet, as carnivores eat more meat and more protein than herbivores do, and a difference in the composition of the amino acids in that diet, as plants and animals contain different ratios of amino acids. What I have done is created a streamlined pipeline. There is existing work that has developed the underlying math and functions behind performing these analyses, but until now, they've only been applied to individual phenotypes. Recently, the Xenomia dataset has provided a multiple sequence alignment for hundreds of mammalian genomes, which means that there is now a very wide range of species and phenotypes that this can be applied to. And what my work has been is creating a streamlined pipeline which can quickly apply these analyses to any phenotype with, as I mentioned before, a multiple sequence alignment and the phylogeny across the tree. What it does is it goes from here, as I mentioned, mapping of a phenotype across a phylogenetic tree, to detecting selection on individual genes in those species, to correlating that selection with a phenotype, and finally, correlating the phenotype with selection on pathways as a whole. As I mentioned, this works on the principle of detecting selection on genes, which means obviously you need a way to do that. The way that we do is via something called relative evolutionary rate. This comes from a package, RER Converge. I have a link down here, which is the underlying work I mentioned that I'm building upon. The way that it functions is by comparing the evolutionary rate of individual genes as opposed to the genome average. If there's no unique selection on one gene, and it's evolving as every other gene in the genome is, we would expect it to evolve at the same rate as the overall genome. So we detect selection by finding genes which are evolving at different rates from what the rest of the genome would predict. You can see here this tree, which is based on the overall genome average. with the various species clustering as you would expect, fairly tightly based on relatedness. This here is a tree based on an individual gene. And as you can notice, there are some branch lengths which are dramatically different between the two of them. For instance here, the Pacific walrus we see is much longer in this individual gene than in the overall genome. This means that this gene is changing much more rapidly. What we can then do is plot this difference between the evolutionary rate of an individual gene against the overall genome. As mentioned, if there is no unique selection on an individual gene, we would expect it to evolve at the same rate as the genome. Here we are plotting the genome average, so the overall genome, against the individual gene. And if there's no unique selection happening, we would expect it to follow along this dotted line here of a 1-to-1 ratio. But as you can see with the Pacific walrus, for this gene, it is evolving much faster in the Pacific walrus than this line would predict. What we can then do is measure the difference of the gene in each species between its value and the line, and that gives us the difference in relative evolutionary rate. We can then directly plot that relative evolutionary rate as you see here. This is showing how fast this gene is evolving in each of the species. This entire plot is one gene with each point representing a species that gene is being expressed in. This analysis is then run repeatedly for all of the genes in the alignment, in our case, over 15,000. What we then do is connect this signal of selection to phenotypes. We can broadly see here in this plot with the carnivores in orange clustering over here on the right, and broadly the herbivores over here on the left. What we then do is we can compare these distributions directly. Most genes are going to look like this, where there is no difference between the two phenotypes. This is the vast majority of genes, and this is a null. There is no difference between the phenotypes. What we're interested in, though, is the genes which have a difference in the distributions. Here you can see one gene where it is evolving slower in carnivores than in herbivores. This typically indicates purifying selection and suggests that this gene is important to the phenotype. Mutations are broadly detrimental, which means that as a gene contributes more to fitness, it is less amenable to mutation. Breaking the gene has a bigger effect the more important the gene is. So genes critical to a phenotype experience increased purifying selection, and therefore tend to evolve slower. Finally, here we see a third plot which matches the plot on the right. All of the data that you're seeing here that is in orange and green is actual data from this analysis. In this case, we see this gene is evolving faster in carnivores, and this can indicate one of two things. Either a direct opposite of what I just described, a relaxation of purifying selection, or it could indicate positive selection. This alone doesn't tell you which, but there are downstream analyses that can be performed to distinguish the two of them. Finally, the last step is by taking these connections between genes and phenotypes, we can then aggregate those to connect whole pathways to phenotypes. What we do is we have these, which are gene ontology geoterms, and we look for those which are enriched for genes under selection. So what we do is we can see if, for example, organic acid catabolic processes, if many or most of the genes in this pathway are experiencing the same selection, we can then connect that selection. We can then connect selection on the pathway as a whole. So, for example, if most of the genes in this pathway are evolving slower in carnivores, we can say the pathway as a whole is evolving slower in carnivores. Now that I've run you through what the analysis is, I'm going to go through what we found in our application to Carnivory. Firstly, our expected findings were validated. As I mentioned before, the most direct change to diet is differences in food composition. We expected to see amino acid metabolism pathways evolving slower. As I mentioned, phenotypes that are more critical tend to be under stricter purifying selection and evolve more slowly. With an increase in peptides in the diet, we would expect to see increased importance of peptide metabolism. We see this pattern in two places, both very broad pathways and specific pathways. So this ranges from organonitrogen compound catabolism, which is really broad, to cysteine metabolism, which is about 3 genes. The broad pathways are highly significant and compose very large gene sets and really broad pathways. In fact, this analysis can be used using a gene set which is based on expression in individual tissues to examine even at the tissue level beyond the pathway level. And in this case, we saw an enrichment in the liver, where much of this amino acid catabolism occurs. We also see very specific pathways. These have really strong associations. This plot here has an association coefficient of about 0.3. Each of these specific pathways are even more dramatic than that. Though, because they have a small gene set, they do not have nearly as high significance as the broad pathways, simply due to smaller sample size. The gene set is smaller, there is therefore a smaller sample size. So we found what we were expecting to see. Amino acid catabolism is conserved. In addition, we found something that was surprising and new, which is that increased evolutionary rate of muscle function in carnivores. As I mentioned before, this analysis can be applied at the tissue level, and we see that here in two different places, both skeletal muscle and the heart, cardiac muscle here in the left ventricle. Again, as before, we also see this occurring in both broad pathways, such as muscle contraction as a whole, and very specific pathways, such as the skeletal muscle thin filament assembly, with the same pattern of high significance and strong association occurring here. As I mentioned before, this may indicate either a relaxation of constraint or positive selection. At the moment, we don't know. We have further analyses that can be performed to distinguish between the two of them, but that's a future direction. In conclusion, to wrap up, I have developed a streamlined pipeline to rapidly analyze multiple phenotypes using existing methods. We have found that amino acid metabolism genes are more conserved in carnivores, and muscle function genes are evolving faster in carnivores, both relative to herbivores. In the future, I hope to apply this pipeline to new phenotypes, and as mentioned, I hope to use those downstream analyses to distinguish the cause of muscle evolutionary rate increase. Finally, I'd like to wrap up with my acknowledgements. I'd like to thank my advisor, Dr. Meyer. She's amazing and very welcoming. Kitty Foley, who did legwork on collecting phenotype data, and all of the undergraduates in our lab. I'd also like to thank our collaborators, the Xenomia Consortium I mentioned, who generated all this data, as well as Irene Kaplan, Daniel Schaffer, Emily Puckett, and Matthew Pollard for their correspondence and advice. And with that, I am happy to take